This video will review the electron transport chain. The electron transport chain takes place within the mitochondria. So if we zoom in on a little slice of the mitochondria and draw it out bigger, this would be our inner mitochondrial membrane. This is our biphospholipid membrane, so we'll give it a little thickness here. Our inner mitochondrial membrane. This means that the top half of this slide, if we draw kind of this space here, this is going to be our intermembranous space. Capped off by the outer mitochondrial membrane. In this drawing then, the whole bottom image is going to be mitochondrial matrix, the innermost portion of the mitochondria. So we draw this positioning out because the electron transport chain spans the inner mitochondrial membrane. And we have these protein complexes, complex 1, 2, 3, 4. The last complex, which is the ATPase, we'll call this the FOF1. It has a very specific job in the story of aerobic metabolism. So within this matrix is the TCA cycle, raring away, taking acetyl-CoA, joining with oxaloacetate, and producing electron carriers such as NADH. NADH is then free to float right up here to complex 1. The job of NAD is to carry electrons from one place to the other. The electron is carried on this hydrogen, as a hydrogen has one electron and one proton. So when NADH gets to complex 1, it can give its hydrogen away and leave, quote unquote, empty, able to go and accept a new electron. So it could then head back to the TCA cycle, grab another electron, and repeat this path. When the NAD drops its hydrogen off in complex 1, complex 1 has the ability of ripping just the electron off of that proton and spitting the proton, the remainder, into the intermembranous space. So throughout energy metabolism, we've talked about moving electrons as moving hydrogen. And this point in complex 1, we're actually able to strip or rip the proton and the electron apart from one another, trap the electron in complex 1, and spit the proton into the intermembranous space. The electron is then passed along the protein complexes. The electron that was dropped off in complex 1 gets transferred over to complex 2. When the electron moves, it moves into a lower energy gradient, and some of the energy is used to pull another proton from the matrix into the membranous space. That electron that's stored in complex 2 is then passed to complex 3. That energy of accepting that electron and moving it into a lower energy state uses some of that energy to pull another proton into the intermembranous space. Complex 3 can then transfer the electron to complex 4. Complex 4 can use some of that energy to pull another proton across. You can see we're generating this big proton gradient. We are collecting protons in the intermembranous space. So there are more proteins outside of the inner mitochondrial membrane than we find protons within the matrix. Now, everything is going well up to this point, but complex 4 has a problem. It is holding an electron, and it's unable to accept another electron from complex 3 until it gets rid of the electron it has now. but there isn't another place for it to go. So complex 4 is going to need something to accept this electron, which is the role for oxygen. This is the only place in energy metabolism where oxygen is playing a role, and it plays the crucial job of being the final electron acceptor. If we look at the reaction that takes place at complex 4 will ultimately take 2 electrons, it will take 2 protons, and it will take 1/2 of an oxygen and produce a water molecule. Perfectly neutral waste product of energy metabolism. So as long as oxygen is here, oxygen will accept that final electron, creating water, freeing up space for the next electron to get passed down the chain. You will notice that up to this point, we haven't yet produced any ATP. What we have produced is a proton gradient that allows there to be a huge difference across the inner mitochondrial membrane. FOF1 allows the protons to fly down their concentration gradient through FOF1, which actually takes this whole complex and spins it. And the spinning of FOF1, that energy is used to take a phosphate and stick it onto an ADP to create ATP. The energy required to produce ATP is generated from the proton gradient. The proton gradient was generated by the previous complexes as they pass the electrons down the chain. This is the final place. This is oxidative phosphorylation, where we take oxygen and make ATP. It's a highly efficient form of ATP generation. You can make massive amounts of ATP, especially if you compare numbers to other processes like glycolysis or even the phosphagen system, which has a very small ATP yield. The one thing is that this entire process is dependent on the availability of oxygen to take the final electron from complex 4.